Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Achieving Success in Neuroscience Clinical Trials, an Integrated CRO Approach. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending me a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point in time, I'd like to thank MedPace, who developed the content for this presentation. MedPace is a scientifically driven, global, full-service clinical contract research organization providing phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. MedPace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and disciplined operating approach that leverages local regulatory and deep therapeutic expertise across all major areas. Now, it's my pleasure to introduce you to our speakers for today's webinar. And first, I'd like to introduce you to Dr. James Warnoff. He's the Vice President, Medical Department at MedPace. Dr. Vernoff is a board-certified neurologist and internationally known clinician scientist with over 20 years of neurology and psychiatry clinical development experience. At MedPace, he provides hands-on medical leadership to sponsors and internal study teams engaged in neuroscience clinical research. Dr. Vernoff received his BA in biology from Columbia University and his MD and PhD from Emory University School of Medicine. He trained in neurology at the Johns Hopkins Medical School, where he served on the faculty for 10 years prior to transitioning to industry. Now it's my pleasure to introduce you to Danielle Caudell Stamper. She is the advanced clinical practitioner at MedPace, and Danielle is a board-certified advanced practice registered nurse. She has over 8 years of clinical and preclinical neurological research experience as a senior clinical research assistant. As a clinical trial nurse coordinator and regulatory specialist, she was involved in the conduct of over 40 clinical trials. A well-published author with articles printed in multiple peer-reviewed journals, she has presented at national conferences and has experience teaching and training others. Next, I'd like to introduce you to Dr. Scott Holland. He's a Senior Director of Scientific Affairs at MedPace Core Labs. Dr. Holland is a physicist with a PH and MS degrees in applied physics from Yale University with postdoctoral training in image processing at Stanford Research Institute and in MRI physics at the Yale School of Medicine. As a former faculty member at Yale University and the University of Cincinnati, and founding scientific director of the Imaging Research Center at Cincinnati's Children's Hospital, he has over 30-plus years of experience in medical imaging research with over 250 peer-reviewed scientific publications. And finally, it's my pleasure to introduce you to Dr. Serena Allen. She is a principal scientist with a PhD in pharmaceutical sciences from East Tennessee State University and an MSc in medicinal and biological chemistry from the University of Edinburgh, Scotland. She has 10 years of combined experience in the preclinical and clinical research space. At MedPace Reference Labs since 2018, she oversees clinical testing and operations within the ELISA, EIA, UPLC, and specialized biomarker areas. Now, it's my pleasure to pass the mic over to James. So, James, when you're ready, you may begin.
Okay, thank you very much. And so I'd like to welcome everybody to the webinar. I really thought that it's very timely to talk about these kind of laboratories in neuroscience trials. since labs and imaging and other kind of testing have moved into a real central place in our clinical trials. So go to the next slide. So just as a little bit of introduction background, I mean, as you said, I've been doing clinical trials for quite a long time. And you can see on the right there, right? When I started, we used inter-office envelopes to communicate. We did have email, but we were still passing things around for wet signatures. We had NCR, CRFs, and key punch data entry. And when I first joined the industry and I asked to see the data in the trial, they brought me a 2-foot high printout on that white and blue perforated computer printouts and said, okay, here's the data, and I had to look through it. But of course, the other thing besides our technology challenges was that we had real challenges in developing neuroscience drugs. We were pretty good at sort of accidentally discovering pharmacologies and then iterating on those sort of findings. And actually, most of the blockbusters that were on the market as I joined the industry were actually CNS drugs, antidepressants, pain drugs, antipsychotics. Et cetera. But as we moved to new mechanisms, it was really hard to get drugs that went into the CNS that engaged their targets, and our outcomes were just hindered by placebo effect and a lot of variability, and it was very hard to develop them. So once we had an outcome that seemed to be responsive, we really just stuck with it. And go to the next slide. So I'm thrilled that the world has really changed now. We actually now have accelerated approvals of drugs in the neuroscience area, and these biomarkers, these measures of labs and imaging, have moved from kind of a scientific interest, a hobby, something we try out in trials, to a critical part of trials. Now, we've always used standard medical tests for safety, and we tried and tried for target engagement. But now, as I'll show a few examples, we're really getting drugs that we know hit the target, and then we can really understand what their effect is, what the right dose is, dose frequency. Now, it is very indication and sometimes gene-driven disease specific. In some areas, say Alzheimer's disease, we have growing collections of imaging, CSF markers, blood markers to diagnose, follow, look at target engagement of our drugs. But other areas, and I always think of the, the psychiatric diseases here, they really have yet to yield to this kind of science in terms of diagnosis. But when I think back of what we knew about Alzheimer's disease back when I was training uh, and we first started getting MRIs of large numbers of patients with dementia and understanding the heterogeneity of them, I have a firm belief that over the coming decades, psychiatric diseases will similarly start yielding to uh, our biologic neuroscience approaches. And I think the other point is that things are moving. And so biobanking, again, it was let's take samples and put them away in case something comes up, to a real possibility that when your trial is complete, 6 months later, a year later, another group will demonstrate a surrogate, a biomarker that is of value, and you're going to want to go back and look at it. So being proactive and understanding who your patient population was and being able to retest after the fact to further explore and iterate in the next trial is becoming something that's important. Next slide. So just in terms of vocabulary and definitions, these definitions of biomarker and surrogate endpoints have evolved over the years. A biomarker in its broadest sense is any characteristic in a patient that's an indicator of the processes going on, response to exposure of a drug. And blood pressure, for example, is a biomarker. In and of itself, it has no clinical significance other than what it tells us about the underlying physiology and the risk for developing clinical diseases like stroke and heart attack. Um, and biomarkers can be molecular markers, histologic markers, imaging, or physiology like ECG and blood pressure. And it's not, though, a clinical outcome. It's not how you feel. It's not survival. It's not signs and symptoms. In the 21st Century Cures Act, there was put in place an actual FDA qualification, and I think this is, you know, to a greater or lesser extent true of regulatory authorities around the world, in which because of the importance now of biomarkers in actually evaluating or drugs dosed as endpoints, that there's more formal approach to saying what is it, how is it being used, what's the conditions under which it's being used, and then what can we use these markers for? Very much like we always had diagnostics as having qualification, our biomarkers for clinical trials are increasingly being that kind of scrutiny. So a surrogate endpoint, though, is a biomarker which can be substituted for a clinical outcome. Now, often that is a provisional or accelerated approval waiting for the actual clinical benefit, but it's accepted that it can predict clinical benefit. Go to the next slide. So I want to just talk to drive this point home, a couple of recent successes and failures. So go to the next slide. So this is aducanumab's label. So this is the first of the antibodies aimed at removing amyloid from the brain that got an accelerated approval. Of course, now there's been another product approved with really better data, but this was the first. And I think it really illustrates the point well. So this is from the label, and it shows you the change from baseline in amyloid PET. And there's a beautiful, clear dose response showing very clearly that this antibody was doing what you wanted it to do over the course of a year, which was decrease amyloid in the brain. You go to the next slide. Now, it was approved. So this really was not a validated surrogate endpoint at the time it was approved, because other drugs had shown decrease, and there was no real clear clinical benefit. But here, again, this is from the label, just the high dose versus placebo, this admittedly small but clear trend towards clinical benefit from the high dose. And that was enough to get the drug approved. Of course, a lot of discussion after that, but just in recent time, moving from mechanism, target engagement, pharmacologic effect of a treatment to a hint of efficacy leading to approval. Next slide. And of course, tofersen in SOD mutation in ALS is another really strong example where this drug was approved based on neurofilament light chain. And here you can see, this very clear difference between placebo, in which it's stable or slightly rising, and a very clear reduction in during treatment of neurofilament light chain. And so this is a genetic specific treatment, and with a very clear effect on not the mechanism of action of the drug, but actually a marker for neuronal injury, which led to its approval. Go to the next slide. Now, I want to say that it doesn't always work out well, and there are a couple of examples of this. This is a study that was published this year that I think illustrates the point beautifully, which is venglustat for patients with a GBA mutation and Parkinson's disease. So, there was this association between these mutations and risk of Parkinson's disease. And so a couple of different approaches to try to intervene and change the activity of the GBA system. So here you can see on the left in plasma and on the right in CSF a target engagement which is rapid and quite strong, both in the periphery, but also importantly in the CSF. So this treatment is doing what it was intended to do quite strongly. So go to the next slide. Clinically, when you first look at these graphs, every time I look at it, I say, well, look at that. It's maybe it worked. You see a clear separation between placebo and the treatment group in UPDRS. The problem is, and it's a little bit hard to see on the slide, it's in the wrong direction. There's actually a strong trend towards worsening in the treated group compared to the placebo group. So although the treatment didn't work and we're not helping these patients, it's a very good demonstration of how our science works here, where we start with a treatment, we know that it's engaging the target, but we don't know in animals, perhaps the animal models had a strong positive effect, but because of the complexities of human disease, we can't be sure that it's going to work in humans. And the proof is in the pudding, if you will, that you need to still demonstrate the clinical benefit in order to have a surrogate endpoint to know that this marker will predict clinical benefit. Go to the next slide. And actually, this is just from the appendix showing all the biomarkers that they looked at. So on the left, you can see plasma, and then next to that, the GL1 in CSF, which I showed in those graphs, very strong effect. And then all these other markers, just no real effect, except one. Interestingly, there's a trend, as you can see in the middle there, CSF neurofilament light, which is trending worsened in the treated group. I think that illustrates an important point about these markers, which is that if I hadn't have seen the SOD ALS data just before, I would say, well, it's noise, it's nothing. Sure, all the other ones look completely even, and this one's just a little bit, it's just a little trend. But because we have instances, and now multiple instances of NfLs predicting clinical benefit, if you looked at this during a trial and said, NfLs are trending worse, I wonder if our patients are doing better, it would at least cause pause about could this be a safety signal? So as biomarkers get validated for clinical benefit, in some sense also they become useful clinically, just like liver enzymes and CPKs become useful as biomarkers because you understand their relationship to pathophysiology. Go to the next slide. So where are we now? So if you have a surrogate endpoint, it's going to be predicting clinical outcome, but so talking about it may be mechanism specific. And in the Alzheimer's example, the amount of benefit you get from the biomarker change is going to be completely dependent on the disease, the patients you have, and everything else that's going on. So even though the amyloid depletion has a very strong signal, the clinical benefit is there, but it's not extremely large. Again, this is how we progress in terms of understanding the relationship between the biology and the disease. And of course, it doesn't add in adverse events because we're always looking at the balance between benefit and risk when we're looking at whether or not to use a drug in the clinic. And so this is all just biological effect in our biomarkers. It's telling you nothing about off-target effects or other clinical effects that may be not desired even for a pharmacodynamic effect of a treatment. And it's slow and incremental, but actually I should say it's slow and incremental, sort of like evolution, slow and incremental until it's not, because very often it just takes one trial and your first clinically proven treatment to demonstrate which biomarkers, which change with treatment, are actually useful in predicting effect and which are not. And we do look across multiple trials, multiple treatments, multiple indications to see which ones, like blood pressure, like liver enzymes, become very important. We know what they mean because we've seen them in so many different situations, and we have a knowledge base that we can use them. It starts preclinically, but one of the things that it does is it makes our lives more complicated because, again, instead of something that might be interested and the scientists can look at after, these become critical parts of development. for approval, for safety. Go to the next slide. So I think one of the things that we're going to be talking about in the next parts of the webinar are how do you deal with these biomarkers now that they're so important. So you see this is from MedPace's uh, uh, data system. Each one of these boxes is a different kind of data that's coming in or that one is looking at. Everything from drug supply to budget, but also then the adverse events, the clinical input, the data from imaging, from ECG, from safety. It's all got to be tracked, kept, and looked at. And the next slide, I think one of the things that we've learned now is that rather than when we used to use NCR paper and have huge printouts, we are looking at data in real time during trials and understanding who's in the trial, what's going on, where are the safety signals, and how is the trial progressing. And without an integration of all that data, you really can't do that well. And so we've always had a tendency to have our data here and there, and at the end of the trial, sort of put it all together and see what happened. We don't live in that world anymore. And I think there's multiple components from the reference lab, the core lab, managing imaging, ECG, other actigraphy, other things like that, and the CRO managing the sites and all of the data. And for me, medical oversight during the trial to ensure safety and data integrity. There's a couple of other things which we'll talk about at the end, which is that this biomarker data, as we understand it, becomes markers of target engagement, becomes potentially unblinding. And so some people can see it, some people can't, becomes complicated. And actually, as things like NfLs become recognized as being something like a liver enzyme or a blood pressure, our investigators and our patients want to know what does the data look like in the trial, and they can't get it any other way than in the trial. Imaging as well. So for managing the patient at the site, then some of this data from being kind of interest and can be hidden from everyone becomes of clinical interest. So we'll talk about that as well. So that's the end of my presentation. So I'm now going to turn it over to Scott to start to talk about the core lab component of this integration.
Yeah, thanks a lot, James. That's a great introduction to the role of a core lab in clinical trials, and I appreciate your description of the difference between biomarkers and endpoints, very relevant in terms of the imaging component. The next slide talks about these biomarkers and endpoints that are provided from imaging. Imaging has the advantage in many cases of being non-invasive and allowing for a relatively fast, almost real-time interpretation, turnaround time. Because it's non-invasive and many types of imaging don't require ionizing radiation, can be done longitudinally to look at changes over time, efficacy, and can also be very relevant in pediatric applications. Imaging can be used for a number of types of endpoints in clinical trials, including eligibility, safety, and efficacy. And in the next few slides, I'm going to use an example from study with multipolysaccharidosis type 2 or Hunter syndrome to kind of illustrate the role of imaging for providing each of these types of endpoints. Let's go to the next slide. So in terms of eligibility, one of the factors can be either the treatment pathway, intraventricular injection of IP in the case of Hunter syndrome, or in other trials, intrathecal injection, other pathways that require a clear definition by an imaging expert that it's possible to get a needle safely into the injection site. And so images can be acquired from the patients during the screening window, uploaded into a clinical trial management system, reviewed by qualified personnel, technologists, and project coordinators, and finally reviewed by blinded independent central radiologists, who then are able to make an assessment about whether this patient can safely be enrolled in the trial. We can even take that one step further and provide this neuroimaging assessment to a next level eligibility safety committee for review. And again, the clinical trial management system integrated between the core lab and the CRO should be able to provide not only the imaging findings and raw images, but also medical history, other clinical data for presentation to a virtual online meeting of safety and eligibility committee, who can then make their recommendations in an electronic format where those are documented and time-stamped in the system for eligibility decision for the patient. So taking that to the next stage, then the next slide talks more specifically about the safety aspects of utilizing imaging as an endpoint or biomarker in a clinical trial. In this case, being a developmental disorder, treatment needs to be administered early in development, where the size of the brain is still relatively small and developing, and the investigational product needs to be calculated precisely to the volume of the brain parenchyma. So in this case, the images are uploaded into the clinical trial management system. In our case, there is an automated brain image segmentation component that is executed within that system, returning contours of the intracranial volume, the total brain volume, and the parenchymal brain volume directly into an ECRF for each patient and time point, so that calculation of IP can be done precisely related to the parenchymal brain volume to ensure safety and accuracy of dosing. And then finally, the next slide talks about the use of imaging as an exploratory efficacy endpoint in this kind of a trial. What you're looking at on the left side of the screen are brain images at screening 24, 48, 104 weeks into the trial, looking at coronal, axial, and sagittal images that have been segmented and color-coded with volumes of key brain imaging or brain regions that are important in the progression and assessment of the disease response to treatment. And those are then automatically populating an ECRF. Those 4 time points are shown on the right side of the screen, showing the volumes of whole brain, cortical gray matter, cortical white matter, subcortical gray matter, cerebellum, corpus callosum, and other brain areas can be extracted from this automated segmentation. And it's important to mention that for all of these endpoints, the role of a blinded independent central reader is critical. When a multi-site study is being executed, the data could be reviewed independently at each site, getting a pretty wide-ranging result with potential biases entering into the decision about enrollment and safety, depending on how eager an investigator is to enroll a subject. The role of central radiologic review is to ensure that all of the data is evaluated in the same way, in a blinded fashion, without any bias in favor or against enrolling subjects. So the numbers are allowed to then be a decisive component in the decision-making process, not only for eligibility and safety, but also looking at the efficacy of a compound. In our image processing pipelines, we can also submit all of this data to a unified segmentation, normalization, and volumetric analysis so that we optimize the sensitivity to changes over time. The next slide sort of departs from MR imaging as the modality of choice. And MR is a very powerful imaging modality. It uses non-ionizing radiation, so it's very safe, can be used in children, can be used longitudinally, but it also has limited sensitivity in some regards. And so PET imaging provides an opportunity for very targeted and highly sensitive assessment of key imaging biomarkers. And this is an example on the opposite end of the age spectrum with patients with Alzheimer's disease, showing on the right side of the screen PET images from FDG, F18 FDG PET, showing metabolic uptake in gray matter. And then the next row showing amyloid PET imaging. And then the bottom row showing PET imaging of tau PET tracers. And there are FDA approved products on the market now for each of these applications, opening the possibility of using highly sensitive PET agents for assessing eligibility, target engagement, as well as the efficacy of treatment for Alzheimer's disease. And the next slide, I think, takes it one step further. And I was actually really pleased to hear your optimism, James, regarding the future of neuropsychiatric treatment. And I agree with you. I think we're on the cusp of developing targeted agents, understanding neurotransmitters and how to engage them for treatment of neuropsychiatric illnesses. So far, there aren't really any approved biomarkers for psychiatry predicting disease outcome or treatment, but I think we're getting there. And this is an example of a very promising PET agent shown. Images on the right of carbon-11 PET tracer, UCDJ, which binds to synaptic vesicle glycoprotein 2A, which has been implicated in major depressive disorder, post-traumatic stress disorder, and chronic schizophrenia. It's important to note that both for the amyloid tau PET agents, FDG, as well as for the carbon-11 PET agent shown here, we can actually use neuroimaging in a quantitative analysis pipeline to measure standard uptake values or standard uptake value ratios with looking at the uptake in reference to a reference area in the brain to allow us to look at changes over time or changes with treatment. In order for all of this to work smoothly, the next slide kind of illustrates the integrated services that a core lab needs to provide in the context of a neuroscience trial. It begins with having the appropriate imaging expertise across modalities to assist sponsors with optimizing imaging protocols, as well as with the advanced analysis of this data to provide the imaging endpoints. Project team should also have neuroscience expertise, and the core lab should provide the technologists who also understand these images can perform quality assurance and ensure that the data passing through to central review is of high quality. Core lab also participates in qualification of sites for participation in multi-site trials. And this is really important in terms of verifying that sites have the necessary equipment to be used in the trial, that this equipment is appropriately maintained, meets quality assurance standards itself, and in many cases it's also been calibrated for use in quantitative assessments. And then finally, training the sites to upload this data in a de-identified fashion. The core lab also assists with training site personnel, technologists, coordinators, so that images can be captured and transferred to the core lab for quality assurance and central review. I've mentioned de-identification of data a number of times, both in the United States with HIPAA, in Europe with GDPR, Asia, and other regions of the world. There are pretty strict requirements now on the de-identification of the data coming into clinical trials for analysis. The system that we utilize, which James mentioned earlier, ClinTrack, is an integrated clinical trial management system that includes an imaging component, which at the site removes all of the PHI so that that data never leaves the site or the continent where it was acquired. And that does help to maintain compliance with local regulatory authorities. And then finally, the most important step in providing a credible clinical trial endpoint from imaging does rely on the expert central review. And typically this involves board certified readers with neuroradiology, nuclear medicine, or other expertise, and the credentials to be able to review and verify that the data flowing from these neuroimaging pipelines is accurate and the endpoint is trustworthy. The next slide, I think, is our last one, just kind of illustrating this integrated pipeline for image workflow from sites to the clinical trial endpoint. So again, providing a clinical trial management system that de-identifies data, allows that data to be uploaded easily from the sites to the core lab, where it can be analyzed and archived, as well as performing QC. In this case, the center column shows an illustration of this automated brain segmentation and returning of these brain image volumes to an automated ECRF for approval by the central radiologist prior to transfer of the data to the sponsor in a format that's been agreed upon in a predefined data transfer agreement. Our core lab also stores this data for 15 years pro forma to ensure compliance with regulatory expectations. And with that, I'll pass the baton to my colleague, Serena Allen, who will go into the role of the central lab in neuroscience trials.
Right, thank you so much, Scott. So now that we've discussed imaging considerations and the pathway from image production to trial endpoints with Dr. Holland, I would like to move forward into fluid biomarkers. So in the past, when we thought of central or reference labs, the first thing that really came to mind was routine chemistry, serology, and other safety screening tests. But with the integrated and specialized approach we've been discussing, and with the increased research, they can now offer much more. In the present day, central labs provide disease-specific biomarker support, which is possible because of new technologies and research that have brought significant advancements in the determination of sensitive and specific biofluid markers. And really, the beauty of this is that a single biomarker can answer more than one question and serve several purposes. For example, specific biomarkers for neurodegeneration and other CNS indications are utilized to confirm the mechanism of action and downstream pathway modulation, improve the diagnostic workup of these diseases, allow for treatment effect monitoring, allow for repeated longitudinal tracking of disease progression over the course of clinical trials, and potentially serve as surrogate biomarkers that can show positive effects of treatment from downstream events. Additionally, biofluid analytes, along with molecular pathology, allow for disease differentiation and classification. Next slide. So now we'll discuss some technologies that have aided in the advancement of CNS biomarkers and have also accelerated drug development. Previously, quantification of many soluble CNS proteins, both in circulation and found in the CSF, has been difficult due to sensitivity limits of the available assays and the analysis platforms. What we were seeing was that the quantification limits of these assays did not really allow for a longitudinal tracking of these proteins at the expected disease range levels. So more recently, ultrasensitive immunoassay platforms have been developed and commercialized for central lab use. Two of these are the Simoa assays from Quanterix and the SPLIC technology from Mesoscale Discovery. The Simoa assay is a bead-based digital single molecule array where each disc microwell can hold one bead. Fluorescence is the detection system on this platform, and the fluorescence of each well can be determined. Now, the MSD SPLIX assay is a 96-well plate-based assay that utilizes a turbo boost label detection antibody coupled with a TurboTag electrochemiluminescent label to allow for sensitive detection. So with these ultra-sensitive technologies, femtogram concentrations are detected, and both single and multi-analyte panels are available. Another benefit is the low sample volume requirements, with some 9-analyte panels requiring only 100 to about 200 microliters of sample. But perhaps one of the most beneficial outcomes from their enhanced sensitivity is the ability to quantify CNS biomarkers in peripheral blood samples. We can move on to the next slide. Now, I want to dive into the importance of the ultrasensitive capabilities and how we can utilize blood-based assays. First, and most importantly, peripheral blood collection is less invasive, especially when compared to lumbar puncture, and with that decreases patient burden. Also, due to this less invasiveness, you have the ability to collect and test for markers at more frequent time points for longitudinal tracking, and also to utilize sites outside of specialist areas. Additionally, these non-invasive tests have proven to be extremely useful in measuring early and specific CNS markers that can differentially diagnose diseases early on in the course of symptom presentation. More specifically, blood p-tau 181 has been used for the prediction of tau and amyloid beta pathologies. Blood p-tau 217 has been used for Alzheimer's discrimination with a 300 to 700% increase in symptomatic Alzheimer's and the ability to differentiate from Alzheimer's from non-Alzheimer's diseases with an accuracy similar to CSF p-tau and tau PET imaging. Blood neurofilament light chain has been utilized as both a treatment response marker for multiple sclerosis, with reductions associated with drug effectiveness, and as a predictive biomarker for the clinical progression of MS. Additionally, blood neurofilament light chain has also been used in the discrimination of ALS from healthy controls. This goes into play what Dr. Vernov was discussing, and is a potential marker of disease progression, as a retrospective study analysis found that it is elevated 20 times higher in ALS patients versus healthy controls. With all these, blood p-taus and neurofilament light chain specifically are now being written in clinical protocols as screening assays, efficacy endpoints, and to confirm drug target engagement. Next slide. So now I want to briefly discuss some genetic biomarkers for CNS studies. The gene mutations or variants that cause phenotypic expression of a variety of these CNS disorders are often deleterious mutations and allele isoforms that can be confirmed and screened for study inclusion through genotyping. So this would include Sanger sequencing, next-gen sequencing, and copy number variant analysis. Now I've listed below some of the major genetic markers for Alzheimer's, Parkinson's, and ALS. For early onset Alzheimer's, we're looking at mutations in amyloid beta precursor protein and the preselin 1 and 2. For late onset Alzheimer's, the most common molecular assay is the ApoE genotyping, with emphasis on the E4 allele, with increases in the number of E4 copies corresponding with an increased risk of Alzheimer's. While Parkinson's is mainly due to sporadic disorders, a variety of genes have been linked to the disease. So roughly 15% of Parkinson patients have a family history caused by mutations in the leucine-rich repeat kinase 2, PARKN2, PARK7 or DJ1, glucose ribosides, and the PTEN-induced putative kinase 1 genes. More recently, RNA-based biomarkers or microRNA have been found as potential markers of Parkinson's. These include microRNA 7 and 153, which modulate the alpha-synuclein mRNA. For ALS, the two primary genetic biomarkers we see are mutations in the superoxide dismutase 1 and variants in the chromosome 9 open reading frame 72. Next slide. So I know we've been focused on biomarkers, but I want to shift gears a bit and discuss the importance of a full-service global central lab for study support and present some considerations for a central lab that come with an integrated CRO model. So first would be scientific expertise in the indication, the analyte or analytes being tested, and the testing methodologies. If method validations for esoteric markers and method transfers are required, you'll want to have a scientific team that's accessible and will work to adapt these assays to be fit for purpose and are familiar with validating assays in a GCP, CAP, CLIA environment. Second, with the complexity of these trials, you want operational excellence, experienced project management teams, and the availability of global PM teams to ensure all sites have around-the-clock correspondence. Third, data management, so real-time data demographic cleaning, daily site reports, web portals for online access to results and custom study reports. And this is where an integrated approach proves most beneficial, as the medical monitors, the sponsor team, CTMs, and other unblinded study staff may access lab reports and data all in real time. Fourth would be logistics management, the ability to build and distribute customized kits, experience in managing global couriers, experience in exporting dangerous goods, and with custom requirements. Fifth would be the biorepository and biobanking services, global availability of these services to ensure sample shipments from North, South America, Europe, Asia Pac, and all of the regions can be stored properly. And you want to make sure these storage systems are monitored 24/7, 365. And of course, as we've discussed, the ability to perform advanced lab testing and analytic capabilities within the expected turnaround time windows. Now I'm going to focus more on the bottom two of this to wrap this up, because at the end of the day, we are talking about sample analysis and producing usable data for clinical studies, and we can't really produce quality data without focusing on sample integrity. So next slide. There are a few things to consider here, and there's a lot more to the process than just shipping and storing when we're talking about logistics, biorepository, and biobanking. First, we'll look at pre-analytical considerations that are surprisingly more often than not afterthoughts. This will include knowledge of the analyte stability during sample collection. Can it be drawn and held at room temperature until centrifugation? After centrifugation, can it be stored at room temperature during the aliquoting process and for a period of time until staff can get samples to the refrigerator or freezer? Does it need to be centrifuged at 4 degrees, kept on ice during aliquoting and frozen immediately? Does the analyte itself require the addition of a protease inhibitor or other stabilizer? Are the sites equipped with all the necessary processing and storage systems? These are questions that need answers. So moving onward to sample collection and shipping, this includes providing easy to follow, standardized collection manuals for both blood and CSF, maintenance of sample integrity and results interpretation. So for example, specifying a traumatic tap, site training materials for standardized PBMC processing and DNA isolation if required at that level, and for the preparation of biopsy samples. Again, here, analyte stability is to be considered for shipping temperatures. Ambient is obviously most cost-effective and utilized where possible, but oftentimes samples require shipment on dry ice or liquid nitrogen, the frequency of shipments from sites to the lab, and an extensive courier network to ensure on-time delivery. Now, lastly, once we have our samples collected, shipped, and at the lab, they will either need to be analyzed or stored. Analysis schedules should be defined by both the sponsor, but also the analyte stability. Batched analysis for manual plate-based assays and complete subject sets where possible to mitigate intrinsic plate-to-plate variability, the availability of long-term storage for future biomarkers and backup aliquots, and potentially long-term stability data for future biomarker samples. So thank you very much. That is the end of my portion of this webinar, and I will now pass it over to my colleague, Danielle, who will discuss some operational and logistical considerations from a CRO perspective.
Thanks, Serena. I've put together a couple case studies, and I just want to illustrate the points that we've just heard from James, Serena, and Scott. As a basis for this, I'm going to work through the conversation surrounding the question, should sponsors disclose individual results from testing performed during clinical trials? And if they do decide to disclose it, should they give it to the investigators or to the patients? The available data on this topic is mostly subjective, but I think that's an even bigger reason to have a team around you to evaluate the difficult choices that you'd come up with during this process. So just starting at the beginning, why do we do a clinical trial? It's to gather data about a representative population and then generalize to a larger group. So the traditional viewpoint on this was that any test done during that process, especially anything that a sponsor paid for, was strictly to populate the data set. So research tests were used for research, and if an investigator wanted to put their medical cap on and get information about their patient, they had to run their own separate tests. So what's really changed? The brain used to be uncharted territory, a black box. As James said, the biomarkers in these early trials weren't usually anything a clinician could really use, but that's not the case anymore. As Scott and Serena have showed us, it's possible to visualize brain function and quantify what's happening at a molecular level, and consequently, PIs want access to this data. It's really exciting for them to have biomarkers that can aid in this direct patient care. So on the next slide, we'll look at our first case study, just a really clear-cut example. So this was a clinical trial. It was a non-inferiority study that looked at the ability of MRI versus CT head to detect a complication post-stroke. And just to circle back to the point about the rapid pace of development, if we look back 6 decades, there were very few imaging modalities for head, and they really had limited utility. It's been, just since the 70s, really the late 70s, that CT scans of the head really became, widely useful. And it's been less than 40 years that MRI was available, at least in the US, at many places. So as Scott showed us, every day those tests are getting better. So if we go to our patient there on the left, they're hospitalized after a stroke. They've agreed to join the clinical trial and are enrolled. And this is a really simple trial. So the patient's just going to get an MRI and a CT head at a really close together time point at the site there at the hospital. And these images are going to be de-identified, really important, by the site. They're going to be packaged up electronically, and they're going to be pushed to that central reader, which we know is going to help reduce, variability and bias. And then, of course, that's going to be data. That data is going to be analyzed. So while all that's happening, the clinical care team is continuing to evaluate the patient. And if they decide in that same time frame that a CT scan of the head is needed, then the patient has to get, if the patient had to get a second scan, they're going to get a bill for that second set of imaging. They're going to go through double the radiation exposure. They're going to have to wait for that second scan, and ultimately that can delay their care. As we all know, time is brain. And meanwhile, the hospital CT scanner is getting clogged up by all these patients who need 2 CT scans back to back. So the study experiences no obvious repercussions if they are willing to share those images with the clinical care team, and have that clinical care team use their local radiologist to read those images. And obviously the site and patient burden is really reduced by this. So on the next slide, let's look at how these studies have changed the conversation. As we said, it's really difficult to imagine in that case study why you would withhold those test results. The test is meaningful in a medical way. The care team will be able to use those images to treat the patient, and the test is clinically validated with very clear-cut ways to interpret the results. So what would we do in the same scenario if it was some new kind of test that hadn't been clinically validated? What if you disclose the results to the patient and it might have a negative impact on the trial itself? Should the sponsor release results in studies where they know that the investigator could become unblinded? The only reason the tests are being done in this trial, is for the study. So if the trial outcome is going to be biased by releasing the results, so why would you even do the trial? So if you can avoid that by having a separate unblinded care team that is involved in disclosing those patient results to the patient, then who would bear the cost for that extra care team participating in the study? And really, what should the clinicians tell the patients about the test results if they aren't clinically validated? How do they apply those results to the care of that patient? So let's look at some of these complicated issues on the next slide in this case study. And as you know from examples that Serena, Scott, and James gave, biomarkers in Alzheimer's disease is research in that is a really hot area right now. So in this case study, the sponsor has a drug, or we call it drug X, and they think it's going to be beneficial in very early Alzheimer's disease. So you can see there in the top left box, our PI enrolls a patient who has really mild cognitive impairment for screening. And thanks to the biomarkers, many of them we've already discussed, the sponsor can predict with some certainty who has mild cognitive impairment due to Alzheimer's disease, and who has other conditions causing that symptom. So you can see in the next box, the patient has a battery of screening tests. They have, the primary investigator gives them clinical outcome assessments. Those are entered into a database, and a lumbar picture collects CSF, and that's sent off for analysis. The patient gets a PET scan to look at those biomarkers like Scott showed us. And they have an MRI head to rule out some other conditions. Blood's drawn, and the serum labs are measured. And one operational consideration I'll point out here is how much site burden there is if separate vendors are used for each part of this process. Besides the site having to schedule all these tests in a really narrow time window, and all the other work that we know is involved in this, teammates from the site, the CRO, and the sponsor have to get trained on every single computer system involved for each vendor. They have to get a username and password for all these separate sites. And I don't know about you guys, but with the security of some of these passwords, my passwords are 15 characters long. The site teams have to enter data into all these different systems. They have to remember, where to send samples and images and data and where to get the test results back. And the CRO and site teams have to keep track of all these vendors' contacts and ensure that the sites ensure that the results are delivered in a timely manner. So having an integrated approach really means that everything goes to one place. All the computer systems can be housed in one umbrella. If you need help, you just call one team of people. So that can be really helpful to relieve site burden and reduce the complexity of this, and increase the speed with which the research process is done, and also to improve patient safety, because any of those labs, any of that process is going to be looked at in the fastest way possible, with all that being integrated into one process. So in the next step, you'll see here that enrollment criteria isn't met, the patient fails screening, the patient is alerted to why that happened. Sorry, if you could go back one slide. So example, the amyloid beta is out of range, then they'll be alerted to that. But the PI is not going to be given a specific number, it's just going to be that it's out of range. And if another medical process is detected, like vascular dementia or thyroid disease, the PI is going to be alerted to that, and they're going to be responsible for distributing that to the patient's direct care team and to the patient, or to whoever's appropriate. So this is the easy choice in our disclosure scenario. You definitely disclose these. But let's look at the next slide. The patient in this scenario is enrolled into the randomized portion of the study. You can see there in that third column, the patient's randomized. They're going to get investigative product or control, drug X or placebo. And then during that process, they're going to be taking that drug. They're going to get repeat clinical outcome measures. They're going to get serum labs checked. And then at the end of the study, they're going to get another lumbar puncture and another PET scan to look at those biomarkers again. So let's say that the investigator asks for the specific value for amyloid beta 4240 or phosphorylated tau. The PIs are savvy. We select the best and the brightest for our sites, after all. They know that 2 decades of research show that these biomarkers are able to discriminate between healthy individuals, those with Alzheimer's disease and other cognitive issues. They know that some of these can predict how rapidly the patient will decline. And they don't have access to labs or test kits all the time, which can get them that information. And in the cases where they are available, sometimes patients' insurance doesn't cover it. So they really want the numbers from us. And should we give it to them? What's the risk of disclosure? So if drug has a clear-cut effect, the PI might be able to tell which patients are in which group, and essentially they're unblinded. That can result in biasing those clinical outcome assessments that they're doing, and that could literally change the primary endpoint. If the drug doesn't have a clear-cut effect, the PI may put his clinician hat on and decide that being in a different study or taking a different drug might be better for the patient, and they might convince the patient to drop out of the study. And then, maybe the patient would have had a drug effect if they had been on the drug longer. Maybe the labs weren't looking different, but the patient was functionally improving. So, it doesn't matter which tests we're talking about, the same questions apply. Another factor is giving the results to the patient. There's no standardized interpretation for some of these. James showed us, for example, that rising neurofilament light is concerning because it's showing neurodegeneration, but which value range is predictive for faster decline in Alzheimer's disease? And even from test to test, which, normal ranges. We just don't know those things yet. So in clinical practice, these test results are now available to patients. Validated clinical test results are available to patients as soon as they hit the electronic medical record. And we know that causes anxiety and misinterpretation. Patients get those test results, they Google them before they see the clinician in clinic, and they've decided that they are dying before they see you and find out that they have nothing seriously wrong. So um, uh, how do we mitigate these risks to the patient? Um, and even if we know what to say to them, how do you make that situation available without unblinding the study team? So do you have an unblinded staff member that's available to counsel the patient? And if that's the case, again, who bears that cost? So let me wrap this up on the last slide here. There's no right or wrong answers. It's a complex process that we just have to work through. This question and others, and having a medical team that's embedded within your operation team and the lab teams that deliver these biomarkers, a really integrated approach is going to ensure that your data integrity is protected, that the study blinding is maintained, so that those endpoints are protected, and that most importantly, the subjects in every trial you do are safe. The integrated CRO approach seamlessly and efficiently will accelerate the pace of this complex neuroscience research while still, maintaining those key points. Thanks for your time. I'm going to hand this back off to Sonia for our Q&A session.
Well, thank you very much, Danielle, James, Scott, and Serena for that very engaging and interesting presentation. I hope everyone enjoyed it. But unfortunately, Danielle, we don't have time for the Q&A, so I just want to remind the audience that you still can send in your questions, but the team at MedPace will follow up with you after this presentation. Now, if you enjoyed listening to MedPace, please mark on your calendars their next webinar, which is slated for Tuesday, September the 26th. So please, I'm going to send you in the link in a few moments' time. The link to that webinar. And I'm also going to send in your link if you have any information or if you require any more further information, resources here, you can use this link. And let me just get that to you here. Okay, so that's the link for further resources. And I'll also send that link for you to register for the up and coming webinar for Tuesday, September the 26th. And there it is, audience members. You should see it now. Perfect. So please go ahead and utilize that. Now, if you have any further questions, please direct them to the email address that's on your screen, and I'm sure the team at MedPace will be happy to follow up with you. So now I'd like to just say thank you very much, everyone, for participating in today's webinar. You will be receiving a follow-up email from XTalks in regards to a survey. So your participation would be greatly appreciated, as it will help us to improve on our further webinars. And now, additionally, you're going to receive a follow-up email from XTalks with access to the recorded archive for this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so they may register for the recording here as well. And as I mentioned before, a survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So now I'd just like to say thank you very much to our speakers for joining us today, James, Danielle, Scott, and Serena, for that very insightful presentation. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.